Good evening, everyone. I'm Gagan Agrawal from Sharad Pawar Dental College. Today, the title of my presentation is Something New, Something Excited. Any guesses? So, by looking at this picture, what all you think? The picture is showing something like brittleness, translucent structure, hard structure. Any guesses? What is it? So, let's have a look once. So, this structure is about enamel. Every one of us know what is enamel. Enamel is nothing but a hard, translucent tissue covering the anatomical crowns of the teeth of man and most of the mammals. Enamel is also called Sustentica adamsia or Amiloblast. From where enamel is developed from? Enamel is developed from a gene that names is CTPI2 that controls tooth enamel production. This is transcription factor which regulates skin and nervous system. Discovered by Krisa QC. Krisa QC is from Oregon State University. So what enamel composed of? It's a structure, no? That something is in that. So what's that? It consists of inorganic matter 96%, organic matter 1%, and water 3%. Among organic matter includes tyrosine-rich amelogenins and non-amelogenins. Inorganic matter includes calcium phosphate, hydroxyapatite crystals, fluoride, strontium, magnesium, and lead. And apart from this, it constitutes 3% of water. Now, what are the physical properties? The physical properties of enamel includes extremely hard like mild steel, so it can withstand mechanical forces. As you all see in this picture, this enamel is very extremely hard like mild steel. Why? It can withstand mechanical forces during mastication, smiling, uh, masticate, etc. Bulk. Maximum bulk around 2 to 25 mm at cuspal region and knife edge thickness at neck region, as you saw in this figure. Specific gravity of enamel is 2.8. Permeability. It acts as a semi-permeable membrane for sea level urea, eye, etc. Color. Light yellow to grayish white depends upon translucency of enamel that is attributable to calcification and homogeneity. Now, enamel structure. What are the enamel structures or what are the structures that is related to enamel? First, enamel structure consists of enamel prisms, enamel rods, prism sheets also known as rod sheet, interprismatic substance that is also known as interrod cementing substances. First, enamel rods. Enamel rods are long slender structures with roughly 5 or 6 sides extending from dentin enamel junction to the structure in wavy codes and oblique direction. The length of the rods are greater than the thickness of the enamel. The diameter of the rods average 4 micrometer along the enamel junction and becomes double at the surface. Sorry. The enamel rods normally have a clear crystalline appearance, permitting light to pass through them. They appear hexagonal, round, or oval, and many resemble fish scales. So, in this, you are looking at enamel rods. These are wavy in courses. Yeah, obliquely directed. Seconds are in permanent teeth, the direction of enamel rods are similar to that of the deciduous teeth at occlusal two-third. But at the cervical region, the root directed rootwise, so the enamel ends cervically as a knife edge. Second is prismatic sheet. Prism sheet is the outermost portion of the rod and takes strains easily because it has more organic substance. It is a substance which cements the rods together. It is approximately 1 micron thick and believed to have proliferated from terminal bar apparatus as enamel globules are developed. This is about interprismatic substance. Submicroscopic structure of enamel. The rods in cross sections has a common pattern, keyhole or paddle shaped pattern with head and tail. In longitudinal section, the heads or bodies in between the tails of adjacent rods would appear like rods are separated by interprismatic substance. So, when we weave submicroscopic structure of enamel, a keyhole or paddle shaped pattern is weaved, as you see in this figure. The apatite crystals appear flat, ribbon-like and arranged with their long axis paralleling the long axis of rods. Some crystals may diverge as much as 40 to 60 degrees from the axial plane of the rods. If someone asks you what are the apatite crystals, what is the arrangement of apatite crystals in submicroscopic structure? So The pipette crystals arrangement is like flat, ribbon-like, and with their long axis paralleling the long arc of the rods. Some crystals may diverge as much as 40 degree to 65 degree from the axial plane of the rods, like in this figure. Now, structural characteristics. What are the structural characteristics of enamel? So, transverse striations, Hunter-Trigger bands, incremental lines of Retzius, enamel lamella, enamel tufts, enamel spindles, enamel cuticle, and pellicle. These all constitute its structural characteristics of enamel. So first we will see one by one. What is first is transverse striations. Lines across the rods with 4 microns interval which becomes more visible when in contact with acid. It marks the daily depositional growth of the enamel rods. More prominent, cross striations occur in a regular period of about every 4 days per 1 week and are known as a stria of Retzius or incremental lines. Transverse striations are also known as incremental line of Retzius. See, when we view microscopic structure, these are the transverse striations, also known as stria of Retzius or incremental line of Retzius. Second is Hunter-Trigger bands. What are Hunter-Trigger bands? Hunter-Trigger bands are alternating light and dark bands originating from dentin enamel junction towards the surface. These are originating from DEJ towards the surface. These are caused by different directions of rods or crisscrossing of rods so that in longitudinal section, some rods are cut transversely, diazone or dark zone, and some are cut longitudinally, parazone or light zone. So, the rods which are cut longitudinally are known as parazone or light zone, and the rods which are cut transversely they are known as diazone or dark zone. Like in this figure, see how Hunter-Trigger bands are developed or Hunter-Trigger bands is seen in microscopic structure. Incremental line of Retzius. These are the brownish lines that mark the growth periods of enamel, beginning and ending layers of enamel. It represents every primary calcification in the enamel. Pericamata and neonatal line or ring. It represents every primary calcification in the enamel. Focus on this statement. Pericamata or it is all neonatal line or ring. Stria of Retzius. See in this figure. What are pericamatas? Pericamata are nothing but indication lines of pickerel. Transfer wave-like grooves that are thought to be the external manifestations of stria of Retzius. Pericamata are nothing but the external manifestations of stria of Retzius. See, in this figure, the both lines of Retzius and pericamata is depicted. See. Now in first figure, healthy tooth with intact pericamata. Now, in second figure, partial loss of pericamata is there. What is neonatal line? Neonatal line is something like ring. So, neonatal line are defined as accentuated lines of Retzius, differentiating the parent prenatally developed enamel from postnatal enamel. Neonatal line is that line which differentiate prenatally developed enamel from postnatal enamel. Present in all deciduous teeth and first permanent molar. This is very important. Neonatal line is present in all deciduous teeth and in permanent teeth it is present only in first permanent molar. This is believed to be the result of abrupt change in the environment and nutrition of the newborn infant. This is in this figure prenatal line, neonatal line and postnatal line. So what is neonatal line? You see as you see in this figure, it differentiates prenatal enamel from the postnatal enamel. What are enamel lamella? Enamel lamella are nothing but leaf-like structure defects extending from the surface of the enamel towards the interior. So, enamel lamella are nothing but a surface structure extending from enamel towards the interior. There are three types of enamel lamella: type A, type B, and type C. So, type A is true lamella. It is hypocalcified, prisms, and interprismatic substance. It is limited to enamel only. What is type A? Hypocalcified, prisms, and interprismatic substance. Now coming to type B. What are enamel lamellae? Type B. Enamel cracks preeruptive can reach dentin, filled with epithelial cell or connective tissue. So type B somewhat reach to the dentin. It is somewhat reach to dentin. And type C. These are enamel cracks Post-eruptive can reach dentine filled with organic content of saliva. So in short, type A is limited to enamel, type B somewhat goes to the up to dentine, and type C totally goes into dentine. Enamel tufts. Enamel tufts are narrow ribbon-like structure composed of bundles of poorly calcified rods and interrod cementing substance originating from dentine enamel junction. The term tuft is used because the structure resembles the blades of grass attached to the parent stem. So, when you look at microscopic structure, you see enamel tufts. It is look like grass-like structure as you see in this figure. What are the enamel spindles? Club-shaped extensions or projections of the dentinal tubules into the enamel. It is an odontoblastic process which extends between the cells of inner dental epithelium before the formation of enamel. This is one of the factors that causes the hypersensitivity of dentin enamel junction. So enamel spindles are one of the factors that causes hypersensitivity of dentin enamel junction. See, this is enamel spindles. Now enamel cuticle and pellicle. What is enamel cuticle? Nasmyth's membrane is a delicate membrane that covers the crown of newly ruptured tooth but is probably soon removed by mastication. Nasmyth membrane is nothing but it is so much delicate membrane that covers the crown of newly ruptured tooth but is probably soon removed by mastication. Pellicle are precipitations of the salivary proteins covering the erupted enamel and may be colonized later by bacteria and food debris. See in this figure enamel cuticle. See Pelican stains. Now second layer you see pellicle appear as a homogeneous layer between plaque and tooth enamel. When plaque and stains attach to your teeth, they don't attach to the enamel. Instead, they attach to the protein pellicle layer. When plaque and stains attack your teeth, they do not touch to the enamel. They touch to the protein like pellicle layer. Scallop dentin enamel junction. The dentine surface on the crown has shallow depressions or pits where the enamel caps are fitted, resulting to the irregular or scallop appearance of the junction of enamel and dentine. This provides a firm attachment of enamel to the dentine surface. So what is the purpose of dentine enamel junction? The dentine enamel junction's purpose is it provides firm attachment of enamel to the dentine surface. See, scallop pattern, DEJ, dentine enamel junction. Now, we talk about enamel structures, enamel characteristics, physical properties of enamel and all and all. But where is from enamel developed? Now, we will see enamel development. Enamel organ originating from the stratified epithelium of the stomodium consists of four distinct layers. Stomodium is the mouth oral cavity, beginning oral cavity stomodium. It consists of four distinct layers. When it develops, it consists of four distinct layers that are outer enamel epithelium, stellate reticulum, stratum intermedium, and inner enamel epithelium. Outer enamel epithelium. Outer cuboidal cells of enamel organ which becomes irregularly producing loops where capillaries are found. It serves as a protective barrier for enamel organ. During enamel formation, cells develop vesicles and villi and a large number of mitochondria, indicating the active participation of cells in the transmission of nutrition. Outer enamel epithelium helps in the participation in transmission of nutrition to the cells. Stellate reticulum. Stellate reticulum, nothing else. These are star-shaped cells in many layers, forming a network within the enamel organ. It supports the production of enamel matrix. It is resistant and inelastic. Therefore, it acts as buffer against physical forces that might distort the conformation of the developing DEJ. It becomes reduced in size, thus it shortens the path of nutrition that comes from the outside. As enamel gets its nutrition from the outer enamel epithelium, but now this stellate reticulum does what? It reduces in size and thus shortens the path of nutrition that comes from the outside. Stratum intermediate, the next layer. More inner compressed layer of flat tube cuboidal cells also supports the production of enamel matrix. The cells show mitotic division even after the cells of the inner enamel epithelium cease to divide. Inner enamel epithelium. Innermost tall columnar cell of enamel organ. It will differentiate into amyloblast that form enamel matrix. So inner enamel epithelium is that layer which will differentiate into ameloblast. What are ameloblast? Ameloblasts are enamel forming cells that form enamel matrix. Cervical loop. When the crown has been formed, the cells of this portion give rise to Hertwig's epithelial root sheet. Cervical loop is nothing but when the crown has formed, the cells of this portion, जो cervical loop के जो portion के जो cell है ना, वो Hertwig's epithelial root sheet को form करते हैं और फिर ये Hertwig epithelial root sheet बाद में root formation में help करता है. Now amelogenesis. What is amelogenesis? Amelogenesis is nothing but a formation of enamel, also called as apposition of enamel matrix. It is a process of development of enamel consisting of two phases. So amelogenesis is constitute of two phases. One is formative phase and other is mineralization and maturation phase. Formative phase is also known as organic matrix formation, and mineralization and maturation phase is the second phase. Formative phase, where there is a formation of enamel matrix. Formation of dentine enamel membrane. The ameloblast deposit a thin matrix extracellularly on the surface of the dentine, uniting with the membrane performativa to become the dentine enamel junction. So this is formative phase. Formation of Tom's process. Now, in the top formative phase, there is a formation of Tom's process. How? The protoplasmic projections of ameloblast into the matrix are the Tom's processes that appear as rows of projections extending intracellularly, responsible for the way the enamel matrix is laid down. Isolation of Tom's process from the cell body of ameloblast by condensation of cytoplasmic substance closely associated with thickened cell membrane called terminal bar, also known as distal terminal bar. Filling in of the distal end of Tom's process with matrix material from the ameloblast. Formation of the fully formed segments of rods or the pre-enamel rods. See in this figure, Tom's process, rod sheet, interrod enamel, enamel rods. Now second is mineralization and maturative phase. The first stage of an intermediate partial mineralization occurs in the matrix segment and the interprismatic substance as they are laid down. The first mineral is actually in the form of crystalline apatite. So enamel is nothing but first mineral is actually in the apatite in the form of apatite crystal. The second stage of maturation starts the height of the crown and progresses cervically. Second stage starts from height of the crown to progress and it will progress to cervically. Seems to begin at the dentinal end of the rods. It begins before the matrix has reached its full thickness. Enamel maturation. Enamel maturation occurs from cusp tip or incisor edge and progress cervically. The maturation of the crystal begins at its dentinal end and progresses to the outer surface. At first parallel to dentin enamel junction and later to the outer surface of enamel. Maturation occurs by growth of the primary crystals till they fuse together. The fibrils between the crystals will become thinner. After eruption, the maturation continues by deposition of ions from saliva to reach 96% of its weight in organic substance. Now, what is life cycle of ameloblast? This is the most important question of all examiners. Yeah, because I am also a student, so I know this comes as LAQ in my DADS question paper in first year. Currently, I am in third year, but this is most important and this is always asked in exam. Always. Life cycle of ameloblast. Life cycle of ameloblast includes 6 stages. What are 6 stages? Morphogenic stage, organizing stage, formative stage, maturative stage, protective stage, and desmolative stage. The short form is MOF MPD. MOF MPD. MOF MPD. Yeah. First, morphogenic stage. Before the ameloblasts are fully differentiated and produce enamel, they interact with adjacent mesenchymal cells, determining the shape of the DJ and the crown. During this stage, the cells are short and columnar with large nuclei that almost fill the cell body. In morphogenic stage, nothing but the outer portion is developed. Yeah. Organizing stage. The ameloblasts interact with the cells of the dental papilla at the periphery, which latter differentiates into odontoblasts. The cells become longer and nucleus-free zones at the distal ends of the cells become almost as long as the proximal parts containing the nuclei. After the first layer of dentine has been formed, the presence of dentine seems to be necessary for the beginning of enamel matrix formation. Now, in formative stage, there are changes in the organization and number of cytoplasmic organelles, and inclusions are related to the initiation of the enamel matrix. Maturative stage. Enamel maturation occurs after most of the thickness of the enamel matrix has been formed in the occlusal or incisal area. Amyloblasts display microvilli at their distal extremities and cytoplasmic vacuoles. This structure indicates an absorptive function of these cells. Protective stage. When the enamel has completely developed and has fully calcified, the ameloblast ceases to be arranged in a well-defined layer and can no longer be differentiated from the cells of the stratum intermedium and outer enamel epithelium. This cell layer then forms stratified epithelial covering of enamel called reduced enamel epithelium that protects the enamel by separating it from connective tissue until the tooth erupts. So reduced enamel epithelium protects the enamel by separating it from the connective tissue until the tooth erupts. Reduced enamel epithelium is very important. It is asked in Viva. Desmolytic stage is the last stage. The reduced enamel epithelium that is formed in previous stage, that is proliferative stage, seems to induce atrophy of the connective tissue, separating it from the oral epithelium so that fusion of the two epithelia can occur. It is probable that the epithelial cells elaborate enzymes that are able to destroy connective tissue fibers by desmolysis. Now, what are the clinical considerations? Now these are the normal of enamel. Yeah, we will see what we have seen till now. Enamel characteristics, enamel structures, physical properties of enamel, how the enamel develops by amelogenesis, life cycle of ameloblast that includes at 6 stages. Now we will see clinical considerations. What in what diseases enamel will suffer? So first question is, how does the knowledge of enamel affect cavity preparation? By looking at this figure, the answer of that question is direct acid etching enamel and bonding properties, restorations and sealants. While we doing restorations or sealants or while doing cavity preparation, it affects affect the cavity preparation. Direct removal of unsupported enamel rods, that is enamel fragile when unsupported. What makes enamel structure susceptible to caries? Crystals dissolved at low pH, acid produced by cariogenic bacteria make enamel structure susceptible to caries. Enamel surface is porous and imperfect, pits, fissures and lamella. How are caries and dental erosion similar or different? Same, both involve chemical insult. Different, caries due to bacterial action, erosion due to environmental factors. At what pH does enamel undergo dissolution? What common dietary substance have a low pH? Answer is pH is range of 5 to 5.5. And what are the common dietary substances that have low pH? The answer is soda prop, sports, drinks, citrus, pickles, etc. The average American drinks more than 53 gallons of carbonated soft drinks each year, more than any other beverage including milk, beer, coffee or water. Pop is a poor dietary choice for three reasons. What accounts for the natural coloration of teeth? What accounts for white spots? Enamel translucent and porous dentin shows through contrast with facial or hair color. Impaired translucence of enamel creates optical white spots. What are some environmental factors that affect enamel formation? How do these occur? Nutrition, systemic diseases like diabetes mellitus, hypertension, thyroidism, high fever, tetracycline, trauma, fluoride, etc. This occurs during tooth development. Tooth development includes 4 stages: birth stage, cap stage, early bale stage, and advanced bale stage. In advanced bale stage, the development of enamel takes place. How does fluoride affect enamel? There are two mechanisms: protective function and by too much affects fluorosis. Protective function indicates inhibitor of crystal dissolution and remineralization with fluorapatite crystals. What are some hereditary conditions affecting enamel? The hereditary conditions that affecting enamel are amelogenesis imperfecta, ectodermal dysplasias, etc. Ectodermal dysplasia is disease of skin. It is a disease of skin. Amelogenesis imperfecta is a condition where there is a disturbance in the formation and maturation of enamel matrix. Enamel hypoplasia, result of the disturbance in matrix formation. Clinically, the surface of enamel may develop pits, fissures or grooves, or enamel may be very thin. Enamel hypocalcification, results of disturbed mineralization of enamel matrix. Clinically, the enamel appears chalky white or opaque and soft. In enamel hypocalcification, enamel appears as chalky white or opaque and soft. So, thank you. You don't have to brush all your teeth, just the ones you want to.